Okay, so again, this is chapter 12 in our lecture textbook. And what we're talking about today is, again, this is our introduction to the nervous system. The nervous system, and some of this, especially this beginning material, is going to be hopefully by way of review because we've talked about a lot of aspects of the nervous system just on our when we were introducing the tissues and going even over the cell biology and transport mechanisms across the cell membrane. But the nervous system is a communication system, right? The nervous system, along with the endocrine system, are the two systems responsible for controlling functions of the body. The nervous system specifically does this by way of sending an electrical signal, the action potential, which we've already talked about when we were for in chapter 10 when we were going over the neuromuscular junction. So again, a lot of this hopefully will be familiar territory. But the neurons, we have two main cell populations of cells that make up the nervous tissue. We have neurons, which are the cells that are responsible for initiating and propagating action potentials, right? And that action potential is the way in which neurons communicate either with other neurons or with muscle or with glands. And then we have what are called neuroglial cells, which are cells that are responsible for supporting and nourishing and protecting the neurons and allowing them, and doing so, allowing them to generate action potentials and work to the best of their ability, so to speak. So, as far as the way in which neurons do their function, right, and therefore the three basic functions of neurons in general, in order for um, in order for the body to work in a coordinated fashion, information has to be taken to the nerve system for processing, right? In order to do this, we have what are called receptors. And so receptors are portions of the neuron, or neurons. We'll talk about the structure of a neuron here in just a moment. But receptors are what we call the dendrites of the neurons that are there to pick up on information in both internally, right? So uh, when you think of the digestive system, for example, you have receptors that feed information to the nervous system, telling the nervous system what's happening as far as whether or not you've had a meal, right? We have stretch receptors that tell the nervous system, hey, the stomach is being stretched, which is an indication that there's food there. We have chemoreceptors. I'm still using the digestive system as an example. Chemoreceptors that tell the nervous system, hey, this, I just ate a meal and tends to be, or it looks to be very high in fat, for example, and there are specific secretions that will be dumped into the into the digestive system in order to aid in the digestion of fats. All of that information internally needs to be fed to the nervous system so that the activity, what we call the motor output, the activity of the body is appropriate for what is going on, right? We also have receptors that tell the nervous system what's happening in the external environment, right? So we have receptors that pick up on temperature changes. If you touch a hot stove, there are reflex arcs and receptors that tell the brain to, well, first of all, the reflex arc will cause you to pull your finger away. And then those pain receptors let the brain know, hey, I touched something really hot and it's painful, and therefore I should do something to care for that now, wound or blister that's going to form. Um, Receptors again are there to collect information and bring that information to the nervous system for the next function, which is processing. So the processing component of the nervous system again is the part of this process in which information that is brought to the central nervous system um, is processed by, for example, the brain, so that you are cognizant of what it is that's just happened. I just use the example of burning yourself. And so the processing in that example would be your brain recognizing that you touched something and that it hurt. Um, and then and I should say actually with the processing and evaluation of information, the processing, so as sensory information is brought to the central nervous system, it is oftentimes, probably more oftentimes than not, brought to the brain, but not always. Sometimes the information that what we call synapse, that information is is transferred from sensory to motor at the spinal cord level. And I bring that up now so that when we get to reflex arcs, maybe you go, oh yeah, that sounds familiar. So kind of more on that to come. Um, and then the last general function of the nervous system, of course, is the effector response. So, as I mentioned a moment ago, neurons communicate by way of an action, using an action potential, by way of an action potential, either with other neurons or with muscles, and all three types of muscles. Or I should say, neurons communicate with all three types of muscles: smooth, skeletal, and cardiac, and then also glands. So the effector response is going to be the response, often the motor response, that again is going to be appropriate depending on the sensory information that came in that initiated that response. Okay, so when for over the next for the next few minutes, we're going to talk about the ways in which the nervous system is organized. So we broadly organize the nervous system into what we call the central nervous system, and the central nervous system is made up of the brain and the spinal cord, and then what we call the peripheral nervous system. And the peripheral nervous system is made up of all of the spinal nerves that leave the spinal cord and go out to, again, all of the tissues of the body that are innervated by the nervous system. When we talk about vernacular and when we're describing entities within the central nervous system versus the peripheral nervous system, the vernacular changes. So, for example, within the peripheral nervous system, we have what are called nerves, right? I just used that word a moment ago when I said spinal nerves. So, what nerves are collections of axons. Axons, as we'll see here in just a moment, are the transmitting portion of a neuron. We kind of have already looked at at least a part of the axon when we were looking at the synaptic knob of a neuron, right? When we were looking at the neuromuscular junction. And again, when we talk about the peripheral nervous system, collections of axons are called nerves. Collections of cell bodies within the peripheral nervous system are what we call ganglia. All right. When we talk about those same entities in the central nervous system, the language changes a bit. So, collections of axons within the central nervous system are called tracts, right? So, a tract is a functionally is the functional equivalent of a nerve. It's just a nerve within the central nervous system. And then collections of cell bodies within the central nervous system are what we call nuclei. So I bring that up now so that when we start going through this material, that language is familiar to everybody. And you can see here, I like this picture. This is from the textbook, but the reason I like it is it allows you to visualize, which again, as we work our way through this material, both in chapter 12 and in 13, and actually in all of the nervous system material, this general schematic is good to keep in mind. So what this is showing you is what's shown in blue is a sensory neuron, and what's shown in red is a motor neuron. And what you can see here, and the reason I really like it, is it's showing you, so this ball right here is the cell body of this sensory neuron. And you can see that the cell body of this sensory neuron is found in the peripheral nervous system, right? This is the cross section through the spinal cord. So this cell body is found outside of the central nervous system. You can see the dendrites here that are, and the dendrites are the receptors, right? So that are found in the dermis. And then in this example, this person is stepping on a pin, right? So that information is picked up by the dendrites of the sensory neuron. It's taken, this collection, we have what's called the dorsal root ganglion, which we'll talk about when we get to the spinal cord chapter. But a ganglion, again, is a collection of cell bodies within the peripheral nervous system. So the dorsal root ganglion is a collection of sensory neuron cell bodies. So this information is taken from the dendrites to the central nervous system, to the spinal cord. And then that information is relayed through what we call an interneuron, which is what you're seeing here in purple. And we'll talk about those more in just a moment, to a motor neuron. Whoops, where's this? Okay, sorry. Um, to the dendrites of a motor neuron, right? And then taken, and then also I like this picture because you can appreciate the fact that the cell body of motor neurons are found within the central nervous system. Again, collections of cell bodies within the central nervous system are called nuclei. And then that information is taken along through the axon and this neuron synapses with the muscle so that so that an effect can occur, right? So I suppose I would like this picture a bit better if this were somebody stepping on a pin and this were a picture of but this could be you pricked your finger and then you pull in response to that. The effect is to pull your hand away from the pin. Okay. Um, so then a little bit more about the way in which we organize the nervous system. So functionally speaking, we organize the nervous system into what we call the sensory branch of the nervous system and the motor branch. So another way in which we describe sensory neurons are afferent. The word is right here, afferent neurons. And as we were just talking about, sensory neurons, and so therefore the sensory nervous system is responsible for picking up on information either in the external environment or in the internal environment of the body and taking that information to the central nervous system for processing. Um, Within the sensory nervous system, we have what we call the somatic sensory nervous system. So this is going to be sensory information that comes from skeletal muscle, voluntary skeletal muscle, right? To the central nervous system for processing. And then the visceral branch of the sensory nervous system is going to include those neurons that are picking up on information that is not under conscious, that you're not aware of, not under conscious control. So when I was using that example a moment ago of the digestive system and the meal that you ate that was high in fat, that information would be taken through visceral sensory neurons to the central nervous system for processing. And then the motor branch of the nervous system is also called the efferent branch, or motor neurons are called efferent neurons. And these are going to be the neurons that are responsible for causing an effect, right? The motor part comes from an activity, motor activity, so motor output. Um, these neurons, again, or this branch of the nervous system, just like the sensory nervous system, is broken into a somatic and a visceral aspect. And another word for the visceral motor response is the autonomic motor response. And we'll be spending at least a good portion of one of the lectures going over specifics of the autonomic nervous system. So suffice it to say for right now that the autonomic nervous system, or what I want you to be familiar with right now, is that the autonomic nervous system has two branches. It has the sympathetic branch, which is responsible for fight or flight types of responses. And then it has what we call the parasympathetic division, which is responsible for rest and digest types of functions. The autonomic nervous system has only a motor branch. So there is no sensory branch of the autonomic nervous system. It is only involved with motor output. And then just like the sensory nervous system, the somatic nervous system is going to be responsible for bringing, or somatic motor neurons are going to be those motor neurons that are responsible for controlling activity that is under conscious control. So these are going to be the neurons that control your skeletal muscles, for example. Okay, so which is what this slide goes over. The somatic motor neurons, again, these are the neurons responsible for allowing you to move your muscles in a conscious, voluntary fashion. And then the autonomic motor neurons are going to be responsible for causing effects that are either parasympathetic or sympathetic in nature, right? So they're either going to be involved with um, rest and digest types of functions or fight or flight types of functions. And as such, these autonomic motor neurons are going to be those neurons that are controlling smooth muscle, which is smooth muscle is not under conscious control, cardiac muscle, and then glandular types of activities. So when I was talking, again, I keep going back to the example I gave with the digestive system, but the digestive system is controlled. Um, there is a branch of the digestive system that, or I guess one way in which the digestive system is controlled importantly is through by the autonomic nervous system. And so when you eat that fatty meal, for example, there are parasympathetic uh, neurons that are responsible for controlling the secretions that would assist in your ability to both break that fat down and then digest it and absorb it within the small intestine. Okay. So, and because I kind of, got a bit verbose on that first slide, some of this information on these slides, the next few slides might be a bit redundant, or maybe not redundant, but is information that I already mentioned. But let's just go over it again. So, nervous tissue again is made up of two broad categories of cells. Neurons are the cells that are responsible for generating and propagating action potentials. Action potentials, we already we've already at the very least been introduced to when we were in the last chapter's material in chapter 10, when we went over the events that occur at the neuromuscular junction and how that those events initiate an action can initiate an action potential in a muscle fiber. So, but just by way of reminder, action potentials have a depolarization and then a repolarization event. These are all or nothing types of, this is an all or nothing type of event. It is, and then the ions responsible for I guess for that are involved in the generation and propagation of an action potential um, are primarily sodium and potassium, right? It's the sodium entering in this case, the neuron that's going to allow for an action potential to occur if we get to that threshold value. And then it's going to be potassium that's responsible for the repolarization, which is bringing the value back to the resting membrane potential after depolarization has occurred. And then glial cells, as I mentioned, are cells, they're not excitable cells, meaning that they're not able to generate and propagate action potentials, but they're absolutely critical in helping, assisting, and allowing neurons to do so. So without glial cells, or when glial cells are not functioning properly, neurons cannot work the way that they're supposed to work. Glial cells, although they kind of tend to get the back seat when it comes to when we talk about the nervous system, glial cells are absolutely integral in allowing neurons to function properly. So they are there to support neurons, to nourish them, to protect them, um, to in at least the case of one, which we'll talk about, to myelinate the axons. They are there. Glial cells are there. The word glial means to support. So these are the cells that support or help the neurons function and allow them to do their job, which is again to generate and propagate action potentials. Okay, so here are some general characteristics of neurons. The excitability feature of them is what we've just been talking about, right? So actually, we've been talking about both the excitability and conductivity aspect of neurons. So neurons are able to respond to stimulation. Um, Oftentimes, that stimulation, as we'll talk about in a moment, is by way of what we call a neurotransmitter. So neurotransmitters um, are able to bind to the dendrites of neurons and initiate a response. And then in response to, in the example I just gave, a neurotransmitter binding, that will cause either the cell to be the neuron to be excited or also inhibited. But the excitability part is telling you that the neuron because of its very negative resting membrane potential, which for a neuron is negative 70 millivolts. It is able to respond to stimulation and then again to generate an action potential. And then if an action potential is in fact generated, that the second characteristic of neurons is that that action potential can be conducted along the length of a neuron. And in doing so, if the action potential, or I should say, when the action potential is conducted along the length of a neuron, it will result in this third characteristic, which is secretion. And by secretion, I specifically mean secretion of neurotransmitters. So neurotransmitters are chemicals, and they are chemicals that neurons use to communicate either with one another or with glands or with muscle fibers. And in fact, we've already talked about one neurotransmitter, and that neurotransmitter is acetylcholine when we were talking about the neuromuscular junction. So some important features or characteristics that I want you to know in relation to this concept. So neurons, for each neuron releases one and only one type of neurotransmitter. There are many different types of neurotransmitters that we will be talking about in this when we go through as we go through this chapter. But it's a one neuron, or I should say one neurotransmitter per neuron type of situation. The neurotransmitter that is released by a neuron can either be excitatory. So, for example, when we were talking about acetylcholine in the context of having a neuron synapse with a muscle fiber, acetylcholine for a skeletal muscle fiber is excitatory, right? We saw that it binds to acetylcholine receptors on the motor end plate of the muscle fiber, and it opens those channels, those chemically gated ion channels, and then that allows for sodium to rush into the muscle fiber, and it brings the resting membrane potential of the muscle fiber from a very negative value of negative 90. And then if we get up to negative 65, which is the threshold value for a muscle fiber, that muscle fiber can generate and then propagate an action potential, right? So in that context, acetylcholine is excitatory for the skeletal muscle fiber. Acetylcholine can also be inhibitory. So the neurotransmitters that are released again for one neuron only secretes one specific type of neurotransmitter, and that neurotransmitter can either be excitatory or inhibitory depending on the target cell, right? So depending on the receptors that the target cell, the target muscle cell, the target glandular cell, or the other neuron that the neuron I'm talking about that secretes the neurotransmitter is synapsing with, depending on the receptors that particular cell has. And then the fourth characteristic of a neuron is this concept of extreme longevity, which is tied to the fifth characteristic, which is that neurons are mostly amitotic. So neurons live. So let me actually touch on this fifth characteristic first. So neurons, by and large, and this is not a hard and fast rule, but by and large, neurons are what we call amitotic, meaning that they do not undergo mitosis to an appreciable degree um, once that human is mature, right? So for the vast majority, and not just for that organism being mature, but in fact, once that organism is born. So for humans, neurons undergo proliferation during embryonic development. But once the human is born, right? Once that fetus is born, and now we have a newborn, the number of neurons stays relatively constant, right? So the number of neurons to an appreciable degree really does not change. What changes and what you see during development and throughout the life cycle of a human or whatever organism you're discussing, but we're talking about humans, is an increase in a change in the number of synapses. And what a synapse is that synapse means connection, right? So we've already talked about a synapse when we were looking at the neuromuscular junction. That was an example of a synapse between a neuron and a muscle fiber, right? So synaptic connections we can form new synapses, and we do throughout the course of our life. And there's tons of new synaptic activity that is formed, of course, as we develop, right? So during infancy into childhood and then into adulthood. And we can still carve, if you will, new synapses as adults, but there's not nearly as the brain is much more plastic, if you will. There's a lot more new synaptic activity that is generated when we are going again from infancy to childhood and then into early adulthood than there is when we are mature organisms. And because neurons are mostly amitotic, they live, that's tied to this concept, the fourth one, which is extreme longevity. So they don't undergo rapid proliferation the way skin cells do, and therefore they live for a really long time. Again, most of the neurons that you have are formed by the time you were born, and for the most part are there, um, you know, up until the point where you die. So neurons, again, they live for an extremely long time, and by and large, they don't undergo mitosis. There is a lot of interesting research that looks at mitotic neurons undergoing mitosis in specific areas of the brain. And I would say, in particular, within the hippocampus, which is involved in the memory formation, the formation of long term memories. We'll get to that when we get to the brain. But this plays an important role. This uh, neurons being mostly amitotic also plays a role in understanding brain cancers and the origin of brain cancers, which we'll get to in a moment. Okay, so when we look at the structure of neurons, um, neurons have relatively large cell bodies. And then I'll just mention right now. So this neuron right here that is being shown in the picture is what is called a multipolar neuron. And the reason I know it's a multipolar neuron is when I look at the cell body, there are many different, if you will, appendages, specific, and those appendages are dendrites that come off of the neuron cell body. And then there's a long axon that can either branch extensively, as is the case here, or not branch extensively, um, depending on the neuron and the situation. But all the same, this is the relatively large cell body of this neuron. Um, and then coming off of the cell body, you're going to have two different structures. You're going to have dendrites, and dendrites are the receiving portion of the neuron. So they are there to receive the when I was talking about that synapse, that connection. Synapses occur between the axon of one neuron, if we're talking about neuron talking to another neuron, the axon, the synaptic terminals, the synaptic knobs of one neuron, and then the dendrites of another, right? So dendrites are the portion of the neuron that receive the signal. And then axons, an axon is the portion of the neuron that transmits the signal. And again, axons can branch extensively. We call the branches of the axon the teledendria. So you can have many different teledendria, or you can have the axon. In theory, you could have the axon not branching at all and just have a one neuron with one synaptic terminal. But more often than not, you're going to see some degree of branching when it comes to neurons. And then I have here more dendrites, more input. What I mean by that is the more dendrites a neuron has, the more information that neuron is able to receive, because the more synapses this particular neuron can have with various neurons. And I guess one other thing to bring up is and I don't think any other good pictures, but I'll try and find some. But when you look at synaptic activity between one neuron communicating to another, or I should say many neurons communicating to one, what you often see with a neuron is that this neuron with all of these different dendrites can be synapsing with, let's say 50 different neurons. You can have synaptic knobs synapsing with all of these different portions of the dendrite. So this one particular neuron can be receiving input from, like I said, 50 different neurons and responding to it. And for those 50 different neurons, those 50 different neurons in theory could be secreting, each one secreting a different, or, maybe not all 50 different neurotransmitters, but you could have, at the very least 20, let's say, different neurotransmitters that this one particular neuron has receptors for. But again, kind of more on that to come I like to give you guys information so that when we get to it, I kind of talk ahead of where we are oftentimes, I know, but that's just so that when we get there, it sounds familiar. Oh, and then one other thing I'd like to point out from this particular image is that off of the cell body, as the cell body tapers into the axon, we have this area called the axon hillock. And physiologically speaking, this is a very relevant area. You can think of the axon hillock as being the trigger. It's kind of like the trigger for the neuron. So when a neuron fires an action potential, the action potential, we'll just say for this class as a blanket statement, the action potential is fired from this particular region of the neuron. If you go on and you take other classes in neurology, you'll find that there are some nuances to that. But for right now, this is where the action potential is fired from. This is the trigger portion of the neuron. Okay. Which is what I have here. So again, the axon hillock is where the action potential is fired from. As I've also mentioned, when we talk about when we talk about neurons communicating with cells, they communicate with either other neurons or muscle cells or glands. And that communication occurs through the axon terminals, also called the synaptic terminals of the axon. So, and specifically these synaptic terminals, and this, I would say the vernacular, just to point out, not that it gets confusing for people, but there are several different ways in which we talk about synaptic terminals, right? So we use the word synaptic terminal, we use the word synaptic knob. Sometimes there will be texts that use the term axon bouton. Sometimes they're called axon buttons, which is what bouton means. But all of those things, they all mean the same thing, which is just to say the end of the axon, and where the portion of the axon that synapses or makes contact with the target cell. I see a typo here. This should say vesicle, so VESI, but all the same, I have on here that they contain synaptic vesicles. So that's really important. This, again, we've already seen when we looked at the neuromuscular junction and we looked at that synaptic knob and those vesicles containing acetylcholine. In that case, we had a neuron with acetylcholine, but no matter the type of neurotransmitter, you always find vesicles for those neurons that are going to be using neurotransmitters, which are most of them. We'll talk about electrical synapses in a moment here, but most neurons communicate using neurotransmitters. So those neurotransmitters are found in vesicles that are located in the synaptic knob. And just like what we saw when we were discussing the neuromuscular junction, the way in which those vesicles release, and this again goes back, kind of draws on knowledge from chapters 3 and 4. But if you go back to when we were talking about exocytosis and endocytosis, exocytosis is the word that we use to describe a cell, a vesicle within a cell merging with the plasma membrane and then releasing its contents out into the extracellular space. So that is the way in which neurons secrete their acetylcholine from the synaptic knob, right? The way in which it happens is the same for all neurons as it was for the one that we discussed when we were talking about the neuromuscular junction, in that you have calcium pumps and voltage gated calcium channels in the synaptic knobs of neurons, and those calcium pumps continually pump calcium out of the synaptic knob to establish a concentration gradient by which you have a lot more calcium outside in the extracellular space than you do inside the synaptic knob. And then when a neuron fires off an action potential and that action potential reaches the synaptic knob, that change in voltage, that's what an action potential is, opens up the voltage gated calcium channels in the synaptic knob. And the calcium, because of the chemical concentration gradient for calcium, will rush into the synaptic knob and it will coat the vesicles, because the vesicles that contain the neurotransmitter and the synaptic knob, those vesicles are coated in a negative charge. So calcium, which has a positive charge, will coat those vesicles, and in doing so, coat them with a positive charge and allow those vesicles to merge with the plasma membrane of the at the synaptic knob. And then in doing so, those vesicles undergo exocytosis and release their neurotransmitter into the synaptic cleft. Um, so that's kind of a preview of what's to come, but hopefully all of that is familiar, because that was what we were talking about when we were going over the events that occur at the neuromuscular junction. Okay. Also within the cell body and throughout the length of the axon, we have cytoskeletal elements, right? So those elements include microfilaments, um, and then what we call neurofilaments, which are intermediate filaments, and microtubules. And what these cytoskeletal elements do for the neuron is they shuttle things back and forth between the cell body and the synaptic terminals. And so that you can kind of think of them, and we'll talk about a couple ways in which substances move between the cell body and the synaptic terminals, but using the cytoskeletal elements is a very fast way in which that can occur. So for example, the the neuron is going to be making its neurotransmitter um, at the cell body, right, through the endoplasmic reticulum and the ribosomes, and then those neurotransmitters need to be taken down to the synaptic knobs, because that's where those vesicles again will merge with the plasma membrane if and when an action potential reaches the synaptic terminals, and so they get there. They being the neurotransmitters and the vesicles get there by way of these cytoskeletal elements, right? Which brings us to transport. So we have, again, two ways in which substances are shuttled around within the neuron. We have what's called anterograde transport. Sorry, you guys, I have a sip of coffee. So anterograde transport is the movement of substances from the cell body, which is where those proteins, for example, are produced, to the synaptic terminals, right? So that's anterograde, meaning forward direction. And then we also have substances that are moved in what we call a retrograde fashion. And retrograde transport describes the movement of substances from the synaptic knobs back to the cell body. Oftentimes, this is for the purposes of degradation. So if there are cellular debris or substances that the neuron wants to put into the lysosome to digest, those substances can be shuttled back to the lysosomes at the cell, which are found in the cell body, via retrograde transport. And this transport can happen either very quickly or what we call slow transport. So I was going to say a little bit slower. So some important, I guess, features or characteristics of fast transport is that it happens in both directions, right? So fast transport uses those microtubules, those cytoskeletal elements. Um, and this is a type of transport that requires energy, and it moves substances in both directions, right? Kind of, you can think of it as a train on tracks shuttling things back and forth. And again, it happens very quickly. Um, and then we have also what's called slow transport. So slow transport does not use cytoskeletal elements. It instead uses just, it's kind of like substances riding the tide of the what we call the axoplasm. So that's the cytoplasm for the axon within the neuron. Um, and in this case, substances, the only types of substances that use that kind of ride the wave of the axoplasm that move in or that are slowly transported are substances that are moved in an anterograde fashion. So retrograde fashion always happens quickly, right? It always uses those cytoskeletal elements. So those are kind of just some important general characteristics of ways in which substances are moved within the neuron. Okay, so now we're going to move on and talk just a bit more about the ways in which neurons are classified. So again, neurons are classified based on the direction that they carry information. So neurons that bring information from the periphery to the central nervous system for processing are called sensory neurons. As I showed you in that picture, several slides back, the cell bodies of sensory neurons are typically found outside of the central nervous system, as you can see here, in the dorsal root ganglion. And most sensory neurons, we'll talk about structural classification here in just a moment, but I'll just make mention of it right now. Most sensory neurons are what we call unipolar. Um, the difference between unipolar, bipolar, and multipolar has to do with structural differences of of the neurons. But unipolar neurons are by and large, I'm sorry, sensory neurons are by and large unipolar. Some are also what we call bipolar. Some of the neurons involved with special senses are going to be bipolar, but most are unipolar. And then motor neurons are neurons that they're also called efferent neurons, and these are neurons that are responsible for motor output, right? So they bring information from the central nervous system out to the periphery to cause an effect. All motor neurons are multipolar neurons, and that's what you're seeing in this picture right here. It was also when I was showing you the example of the neuron a moment ago. That was a multipolar neuron. You can tell it's a multipolar neuron because you have many dendrites that come off of the cell body, and then you have an axon hillock that tapers into the axon, and then the branches, the synaptic terminals of the axon. This is showing you a unipolar neuron, and the way you know it's unipolar by looking at it is that it only has, here's the cell body, and it has one process coming off of the cell body, and then that process branches, and it branches into dendrites or into an axon, right? So, and again, all motor neurons are multipolar. And then another important characteristic of motor neurons is that their cell bodies, for the most part, are found within the central nervous system. Again, you can think back to that picture we went over several slides ago, or if you look here, you can see this is a motor neuron with its cell body within the central nervous system and the spinal cord. And you can also kind of appreciate in this picture that this is a multipolar neuron. And as you can see here, this is a unipolar neuron. Okay. And this is really important. Then we have what are called interneurons. And I feel like interneurons are oftentimes um, described, at least when this information is introduced, as being kind of a footnote. But in fact, interneurons make up, when we talk about neurons, about 99% of the neurons that we have by volume are interneurons. So they are incredibly important. Most of our neurons are interneurons. And what interneurons do is they are responsible for receiving the information from a sensory neuron and then processing that information and then transferring that information to a motor neuron so that an effect can take place, right? So interneurons are the ones that decide, if you will, how your body should respond to the sensory, what the motor output should be, that is appropriate based on the sensory information coming into the central nervous system. Within the brain, the vast majority of the neurons that you have, most of the nervous tissue in the brain, that are most of the, I should say, the neurons in the brain are interneurons. They are absolutely key to facilitating communication between the sensory branch of the nervous system and the motor branch of the nervous system. Most interneurons are multipolar neurons. You can see here's a cartoon or an illustration of one right there. They are found entirely, they're the entirety of the interneurons, so their cell bodies, and then their processes are found entirely within the central nervous system. And they are able to synapse with many different neurons, right? So again, when you think of interneurons, think of them as the ones that receive, store, process, and pass along the information from the sensory branch to the motor branch. Okay, so um, here's a cartoon illustration of the way in which we structurally classify neurons. So we already have discussed multipolar neurons, which again are motor neurons. And then we've discussed sensory neurons, which are unipolar for the most part. We also have, and this is kind of a misnomer, so I'm going to say this right now so people kind of cross this out. This says bipolar interneuron. I want you to think of bipolar neurons as being a special type of sensory neuron. So, bipolar neurons, you can tell they're bipolar because if you look at the cell body, there are two processes that come off of the cell body. That's why they're called bipolar. And these are sensory neurons that are specifically associated with the special senses. So, with smell, taste, auditory. They also can be classified um, as in depending on the way in which you're classifying them, they can be classified oftentimes as interneurons. But I want you to think of them as the sensory neurons that are associated with the special senses. Um, what else do I want to say? Oh, and then pyramidal cells. So pyramidal cells are examples of oh, and I guess before I say that, one other thing, most interneurons are the structurally speaking are multipolar neurons. I think I said that a moment ago, but just good to hear it again. So pyramidal cells are also called anaxonic neurons. These are a special type of neuron. We're not going to spend much time talking about them aside from right now, really. And your book makes brief mention of them, but doesn't go into a lot of detail. So for this, at this point, what I want you to know about them is that they are neurons that don't technically have axons or dendrites. And as such, they don't technically, they're not able to generate and propagate action potentials. What they do is they produce local changes, local electrical changes that are responsible that play a role in a summative way of allowing action and helping to facilitate action potentials and they're in neighboring neurons to those pyramidal cells. But again, I'm not going to test you on pyramidal cells. I just want to draw your attention to the fact that they exist. So if you go on and you do anything in neurology, they are familiar. You've at least heard of them before. Okay, so nerves. Again, nerves and or tracts, depending on the context in which you're talking about them, are collections of axons. We're going to focus on nerves. So these are going to be collections of axons within the peripheral nervous system. And just like what we saw when we were talking about muscles, uh, nerves are and what they're made up of, and the axons have different connective tissue sheets that are there to help insulate and protect and act as an anchoring point for vessels and help to organize the axons and group them into nerves, right? So when we look at a nerve itself, there's a connective tissue sheath that we find that covers the outside of the nerve, and it's called the epineurium. The epineurium is very important in that it provides support and insulation and protection to the nerve. Um, and then the nerve itself, if you were to unwrap the epineurium, you would see that the nerve itself is made up of these different groupings that are called fascicles. Again, that should be a familiar term to everybody. It's just like what we saw when we were talking about muscles. And fascicles are made up of groups of axons that are wrapped in what we call a perineurium, which is another connective tissue sheath. Um, the perineurium in particular is very important in that it acts as an anchoring structure for arteries and veins, so for the vasculature. And then if you were to unwrap the perineurium, you would see that each fascicle is made up of individual axons, and each axon is covered in an endoneurium. So the endoneurium plays a really important role in electrically insulating and therefore electrically isolating each axon from one another. There are also vessels that are anchored to the endoneurium, specifically capillaries, that are responsible for providing the axon of the particular neuron that they come from with nourishment and with gases and bringing oxygen, removing CO2, things of that nature. Okay. Um, so let me go. We'll go through this slide and then I'll end this lecture for the day because we're an hour in. And then we'll pick up tomorrow with where we left off. But as far as the way in which the way in which nerves are classified structurally, we classify nerves into, structurally speaking, into two groups: cranial nerves, which are those nerves that extend from the brain, and then spinal nerves, which are those that extend from the spinal cord. And then functionally speaking, nerves can be, most nerves, I should say, are what we call mixed nerves, which means that they are made up they are made up of axons that come from sensory and motor neurons. Um, Mixed nerves are most of the nerves, the named nerves. So in lab, as you learn the nerves, most of the nerves that you will learn are mixed nerves. And then, but we also have nerves that are just sensory nerves or just motor. And so uh, that's where I'm going to end with today. So we'll pick up tomorrow with synapses.